Hello students, welcome to EPG Pathshala. Today we are going to discuss about structure of fat soluble pigments. I am Dr. Rashida Kanchwala. All of you are well acquainted with plants. As you know that plants are of different colors and their colors are due to color pigments which are embedded within their cells. Today I am going to teach you about structure of fat soluble colors, so as to develop a better understanding in their handling. After going through this module, you will be able to gain knowledge about different fat soluble colors, to know the conditions which affect their composition, to learn to retain them during different processing techniques. The color of vegetables and fruits are due to presence of some chemical compounds within them. As plants can't move around, they have developed procedures to grow, to defend, to protect and to heal themselves. They produce different chemicals to fulfill these requirements. One of these chemicals are plant pigments, which along with fulfilling different plant requirements are cause of delicate and bright colors of fruits and vegetables. They may be present as separate chemicals dissolved in cytoplasm, inside cell wall, in the vacuoles, plastids or crystals within cytoplasm. Pigments in plants are generally surrounded by cell walls that provide an elastic support for retaining the contents of cell. Mature cells also contain vacuoles, which are separate compartments filled with a fluid, cell sap, and are composed of dissolved sugars, salts, organic acids, pigments and other materials. Also located within the cytoplasm are separate inclusion bodies called plastids. Most of the pigments occur in these plastids. A pigment may be present in the cytoplasm as crystals. Sometimes the water soluble pigments are present in vacuoles and not directly dissolved in cytoplasm. The structure of pigments are complex, and as per presence or absence of hydrophilic or hydrophobic chains, pigments can be divided into two categories, that are fat soluble and water soluble. Here we will discuss fat soluble pigments. The chief fat soluble pigments are chlorophyll, carotenoids and curcumin. Let us know their chemistry one by one. Chlorophyll is the pigment which is present in almost all plants, present in all leaves and green stems of plants. It is a green and oil soluble color pigment. They are esters of pyrrole and are derived from porphyrin, a fully conjugated tetrapyrrole. It contains 4 pyrrole groups, 1 magnesium in the center and 2 ester groups. Chlorophyll is essentially made up of 2 parts, a substituted porphyrin ring and a pyrrole, the long carbon chain. The porphyrin ring is an excellent chelating ligand with the 4 nitrogen atoms binding strongly to a coordinated magnesium atom in a square planar arrangement. The porphyrin ring of chlorophyll is also found in heme of hemoglobin and vitamin B12. Heme consists of a porphyrin similar to that of chlorophyll, but with an iron ion in the center of the porphyrin, and vitamin B12 possesses a cobalt iron instead of magnesium. In vegetables and fruits, many different types of chlorophylls are found. All of them possess a porphyrin ring and a side chain with modifications in chain chemicals. The empirical formula of chlorophyll is C55H70MgO6N4 Mg. Different chlorophylls vary slightly in this formula. There are many types of chlorophylls in plants. Chief of them are chlorophyll A, chlorophyll B and B. Both of them are found in ratio of 3:1 in plants. Chlorophyll A has an empirical formula C55H72MgO6N4 and its molecular weight is 893.49. Chlorophyll A is the principal photosynthetic pigment. It exists in all phototrophs with bacteria being the exception. Chlorophyll A is black color in solid state and greenish blue in liquid state. It strongly absorbs light in blue and red region. It is a specific form of chlorophyll used in oxygenic photosynthesis. It absorbs most energy from wavelength of violet blue and orange red light. As it reflects green yellow light, it is visualized as green, which is the color of most plants. Though chlorophyll A is essential for most photosynthetic organisms to release chemical energy, but it is not the only pigment that can be used for photosynthesis. It is true that all oxygenic photosynthetic organisms use chlorophyll A, but differ in accessory pigments. Chlorophyll A has a chlorine ring whose 4 nitrogen atoms surround a central magnesium atom and has several other attached side chains and a hydrocarbon tail. All chlorophylls contains chlorine ring where a magnesium ion is encased in a large ring structure known as a chlorine. The chlorine ring is a heterocyclic compound derived from pyrrole. 4 nitrogen atoms from the chlorine surround and bind the magnesium atom. The magnesium center uniquely defines the structure as a chlorophyll molecule. As other chlorophylls, chlorophyll A has a porphyrin ring with attached side chain. Side chains are attached to the chlorophyll ring of the various chlorophyll molecules. Different side chains characterize each type of chlorophyll and alters the absorption spectrum of light. Chlorophyll A has a long hydrophobic tail which anchors the molecule to other hydrophobic proteins in the thylakoid membrane of the chloroplast. Chlorophyll C is the chlorophyll that occurs only in algae. It has a blue greenish color, particularly significant in its absorption of light in 447 to 452 nanometer region. It is an accessory pigment. It passes on the light excitation to chlorophyll A. It also has a porphyrin ring structure but does not have an isopyrenoid tail or a reduced ring D. Chlorophyll C can be further divided into chlorophyll C1, C2, C3 plus at least 8 other newly found subtypes. Chlorophyll D has an empirical formula of C54H70MgO6N4 and its molecular weight is 895.462. Chlorophyll D is present in marine red algae and cyanobacteria. It absorbs far red light at 710 nanometer wavelength, just outside the optical range. Let me discuss another pigment, that is carotenoids. Carotenoids are a group of yellow, orange and yellow-orange pigments. Carotenoids are hydrocarbons of derivatives of hydrocarbons. They are always associated with chlorophyll in the chloroplast and protects the chlorophyll and sensitive enzymes in plants from destruction against harmful light radiation. The basic structure of carotenoids is an isoprene. Carotenoid molecule consists of isoprene units which contains 5 carbons. Chemically, carotenoids are polyunsaturated hydrocarbons containing 4 carbon atoms per molecule, variable number of hydrogen atoms and no other elements. They may have terminal hydrocarbon rings on one or both ends of the molecules. All are colored to the human eye. Due to extensive system of conjugated double bonds, carotenes are structurally tetrapenes, meaning they are synthesized biochemically from 4 10-carbon terpene units, which in turn are formed from 8 5-carbon isoprene units. We can say that carotenes are polyene hydrocarbons with or without terminal rings. They may or may not have additional oxygen atoms attached. Carotenoids are usually lipophilic due to the presence of long unsaturated aliphatic chains attached to them. As per presence or absence of oxygen, carotenoids can be classified into 2 subclasses, carotenes and xanthophylls. Let me know about xanthophylls. The molecular structure of xanthophylls is similar to that of carotenes, but xanthophylls contain oxygen atoms while carotenes are purely hydrocarbons with no oxygen. The oxygen in xanthophyll is present either as hydroxyl group or as epoxide pairs of hydrogen atoms that are substituted by oxygen atoms acting as bridge. Due to presence of oxygen, xanthophylls are more polar than purely hydrocarbon carotene, and it is this difference that allows their separation from carotenes in many types of chromatography. Xanthophylls are a group of compounds. The chief of them are lutein, zeaxanthin, neoxanthin, boilaxanthin, flavoxanthin and alpha and beta cryptoxanthin. Beta cryptoxanthin is the only known xanthophyll to contain a beta ion ring, and thus beta cryptoxanthin is the only xanthophyll that is known to possess pro-vitamin A activity for mammals. Xanthophylls are found in all young leaves. A chief source of lutein in our diet are papaya, peaches, prunes and squashes. Naturally, most of xanthophylls are yellow-orange colored pigments. Different xanthophylls possess different hues. As lutein can absorb blue light, it appears as yellow color. Zeaxanthin appears yellow-orange color. Cryptoxanthin are other types of yellow-orange colored carotenoids. Alpha cryptoxanthin appears as yellow colored pigment, while beta cryptoxanthin is orange in color. Let me discuss lutein. The chemical formula of lutein is C40H56O2. Lutein is one kind of xanthophyll found abundantly in fruits and vegetables. Although lutein and zeaxanthin are isomers, but they are not stereoisomers. Both of them are interconvertible and are present with each other. So, they are typically reported as lutein and zeaxanthin or lutein plus zeaxanthin, lutein and zeaxanthin. They are not pro-vitamin A carotenoids. Both of them are found in most of the fruits and vegetables. Lutein is found to be in higher amounts green leafy and yellow colored non-leafy vegetables as compared to fruits, but some fruits such as papaya and persimmon have high amounts of lutein and zeaxanthin like that found in vegetables. Watermelon and raw pink grapefruit are also good sources of lutein and zeaxanthin. Numerous studies have identified lutein and zeaxanthin to be essential for components for eye health. They are the constituent of the main pigments found in the yellow spot of the human retina. which protect the macula from damage by blue light, improve visual acuity and scavenge harmful reactive oxygen species. They have also been linked with reduced risk of age-related macular degeneration and cataracts. Lutein is present in plants as fatty acid esters. They are one or two fatty acids bound with the two hydroxyl groups, as they are made up of long polyene chains with conjugated double bonds. They are capable of more light absorption. Therefore, they possess the distinctive light absorbing properties. The polyene chain is susceptible to oxidative degeneration by light or heat and is chemically unstable in acids. Volazanthine and neoxanthine are biosynthesized from zeaxanthin through different chemical reaction. Volazanthine has a chemical formula of cis-40H56O4. The xanthophylls pigment with an orange color is volazanthine. It is found in a variety of plants including pansies. It is biosynthesized from zeaxanthin by apooxidation. As a food additive, it is under the E number of E161E as a food coloring. Neoxanthine has a chemical formula of C40H56O4. It is a major xanthophyll found in green leafy vegetables such as spinach. In plants, it is an intermediate product in the biosynthesis of the plant hormone abscisic acid. Enzyme neoxanthine synthetase converts volazanthine to neoxanthine. The chemical formula of flavoxanthine is C40H56O3. Flavoxanthine is a natural xanthophyll pigment with a golden yellow color. It is found in small quantities in a variety of plants. As a food additive, it is used under the E number E161A as a food coloring agent. Cryptoxanthin bears a chemical formula of C40H56O. Cryptoxanthin simulates with beta-carotene. The only difference between both of them is that cryptoxanthin has got an additional hydroxyl group. In a pure form, cryptoxanthin is a red crystalline solid with a metallic luster. It is freely soluble in chloroform, benzene, pyridine and carbon disulfide. It is found in orange rind, papaya, egg yolk, butter and apples. Let we know about carotenes. These pigments are usually present in chromoplast of the cells. They are found in yellow, orange leaves, vegetables and fruits. Carotene is also found in green leaves, but the prominence of green color of chlorophyll mask is and the orange color of carotenoids can be seen in autumn leaves as the chlorophylls break down. Carotene is a pigment responsible for production of vitamin A in men. It is an absorption spectrum of about 450 nanometer. Carotene acts as an accessory pigment, passing energy to chlorophyll A for use in light reactions and protecting chlorophyll from excessive light and from oxidation by the oxygen produced in photosynthesis. There are many carotenes found in plants. The chief of them are alpha-carotene and beta-carotene. Besides them, gamma, delta, epsilon and zeta carotene are also found in plants. Carotenes contains 8 isoprene units. Out of 8 isoprene, 2 isoprenes are cyclized at each end. The cyclization process distinguishes between alpha and beta carotene. The cyclic ends of beta carotenes are identical. That is, there contains beta-ionine like structure. Thus, oxidative fission of beta-carotene yields 2 molecules of vitamin A. The cyclic ends of alpha-carotene differ in that one is an alpha-ionine and the other one is beta-ionine. Therefore, on fission, alpha-carotene provides only 1 molecule of vitamin A. Alpha-carotene is found in matured green leaves. Alpha-carotene and beta-carotene are found in a proportion of 1:9 in mature leaf. Hydrolysis of alpha-carotene yields 1 molecule of vitamin A. Gamma-carotene has only one end cyclized. The cyclized ring is beta-ionine. Therefore, it also yields 1 molecule of vitamin A. Beta-carotene is the major common carotene in mature fruits and vegetables. It yields 2 molecules of vitamin A on hydrolysis. Beta-carotene is composed of 2 retinyl groups in the mucosa of the human small intestine. It is broken down by beta-carotene 15,15 monooxygenase to retinal, a form of vitamin A. Beta-carotene can also be stored in a liver and body fat and converted to retinal as and when needed. Therefore, it is an important form of vitamin A for humans and some other mammals. The alpha-carotene and gamma-carotene contains single retinyl group. Therefore, although they have some vitamin A activity, but it is less than beta-carotene. Similarly, xanthophylls, carotenoids, beta-cryptoxanthin also possess only one beta-ionine ring and low vitamin A activity. All other carotenoids, including lycopene, have no beta ring and thus no vitamin A activities. These carotenoids, however, possess antioxidant activities and thus they are biological important in other ways. Animal species differ greatly in their ability to convert beta-ionine containing carotenoids to retinol. Gamma carotenoids is found in a small amount in fruits and vegetables. Like alpha carotenoid, it also yields 1 molecule of vitamin A on hydrolysis. Delta carotene pigment is red-orange in color. It does not yield vitamin A molecule on hydrolysis. Now we come to lycopene. Lycopene is the red colored carotenoid. The red color of fruits and vegetables such as tomato, watermelon or grapefruit is due to the presence of lycopene in it. Although lycopene also belongs to the carotenoids like alpha and beta carotene, but the cyclization of both the rings are different. From beta carotene, it does not possess a beta ionine ring, therefore it is not a vitamer of vitamin A. It is an uncyclized carotenoid with open straight chain hydrocarbon consisting of 11 conjugated and 2 unconjugated double bonds. Most of the lycopene occurs naturally in all trans form. The red color of lycopene is mainly due to the many conjugated carbon double bonds, as it absorbs more visible spectrum compared to other carotenes. Lycopene has 2 more double bonds than beta carotene, hence it appears more red. Lycopene pigment is abundant in fruits such as papaya, watermelon, pink grapefruit, guava and apricots. Lycopene is more easily absorbed in our body after cooking. The all-trans isomers of lycopene is the most predominant geometrical isomer in fruits and vegetables. Lycopene has many isomers. 7 double bonds of lycopene can be isomerized to mono, cis or poly cis isomers. Based on the isomeric conformation of lycopene, 5,6 lycopene is the most stable isomer, followed by all-trans and 9 cis lycopene. Lycopene cis isomers are more soluble in oil or organic solvents than all-trans lycopene. While beta carotene appears yellow to orange besides lycopene, astaxanthin is a red colored pigment. Lycopene isomerization occurs in these conditions. under the stimulated gastric digestion, thermal processing in low pH. Heating at 60 degrees centigrade and 90 degrees centigrade favors the isomerization of thermal processing in low pH. Heating at 60 centigrade and 80 degrees centigrade favors the isomerization of lycopene. Isomers of lycopene are generally formed in acidic pH, and 13,6-lycopene is formed as a result of thermal processing. Lycopene is a powerful antioxidant that may help protect cells from damage. Therefore, it is used for preventing heart disease, hardening of the arteries, and cancer of the prostate, breast, lung, bladder, ovaries, colon, and pancreas. Some food rich in lycopene are papayas and oranges. The last fat-soluble pigment is curcumin. Turmeric, a well-known spice, contains curcumin. It is an oil-soluble lemon yellow pigment. This pigment fades in light but has good heat stability. Its light sensitivity can be decreased by the addition of aluminum ions, which form a complex with curcumin and thus protect degradation by light. Turmeric is widely used in curry, soups, and confectionery. The main compounds in turmeric are curcumin and its derivatives D-methoxycurcumin and bis-demethoxycurcumin. The latter differ from curcumin in that the CH3 group are removed. It is not soluble in water but dissolves in oils and alcohol. It is quite stable and gives a bright orange yellow color to products. Lycopene is a powerful antioxidant that may help protect cells from damage. Therefore, it is used for preventing heart disease, hardening of the arteries, and cancer of the prostate, breast, lung, bladder, ovaries, colon, and pancreas. Curcumin are widely used in foods and cosmetics. Applications include canned beverages, bagged products, dairy products, and ice creams, yogurts, yellow cakes, biscuits, popcorns, sweets, cake, icing, cereals, sauces, and many others. Curcumin also acts as an antioxidant and has been added for this purpose to oils and fats. It also protects against rancidity in bakery products such as cakes. Turmeric has also been used as a traditional medicine in India and other Asian countries, but it is not used for this purpose in foods or food supplements. To sum up, plants contains various water-soluble and fat-soluble pigments. The chief fat-soluble pigments, chlorophyll is also the chief photosynthetic pigment in plants. Carotenoids in plants provides colorful hues to them. Besides this, they have biological importance to humans as some of them are pro-vitamin A. The most of them are antioxidants. Curcumin, the yellow color pigment of Indian curries, has many medicinal values in it. We can say that pigments not only colors our platter but also savors our life. Thank you.